Okay.
Cool.
Maybe we
all
should
the
talk
medical
about that.
data. It's,
Yeah.
it's,
Rolling?
it's a big
Okay.
data set.
I'm in a slate.
You're in. Yep.
I'm in.
Yep.
Slate. Okay. Now you gotta let me get by.
We'll let you get by. Were you able to see the slate?
Can I, Drew, can you just
Yeah.
point toward me for just a second? I don't need the clap. I just need to see the slate.
Good.
Yeah, okay.
Good. Okay. Let me get by.
Sure.
Kevin will yell for action. Kevin will let you know when I'm
It's all so natural, isn't it?
seated.
It's
Well,
okay. we try. It's better than we did
whoops.
a lot of stuff. Oh, something is still on.
I think I can do this without getting up.
The advantage of very long arms.
Let's see.
Kevin, we're not getting the computer in the light, are we?
I'm going to get it out of the way in just a moment as soon as I turn
Oh,
it
no,
on.
I meant from where it was.
Oh yeah,
out
maybe
of the
I'll.
way.
Do you mind if I just shut the cover? Yeah, great. Awesome.
What I'm going to do is light down, out of the way. Okay?
Okay.
Great. I'll just shut just for
Okay.
aesthetically.
Okay.
All right.
Alright.
So.
You guys film me that whole time?
Yeah.
We are
Okay.
rolling. And
Whatever. It's
Yeah,
digital.
I just assume
Delete it.
everything's recorded all the time.
Right.
It's the safest.
Yeah.
It's a good assumption.
All these politicians that get their off-color
Yeah,
comments.
but then we know
there's-
sometimes it doesn't even matter. Right?
Oh, well, in the new world it doesn't
Yeah.
matter. Right? 'Cause
Anyway, so you guys
All
ready?
right, we are ready.
We're ready?
Action.
All right. So George, first of all, thank you so much for doing this. I know you're very busy. And we will put on screen all of your amazing credentials, but usually we like to ask somebody to just introduce themselves.
Yeah,
And so
sure.
how about if I just say, go ahead and do that and just say, I'm George Church, et cetera,
sure.
and what you're working on. So go ahead.
Right. So
before you do that,
Yeah.
let me just lower just a little bit.
Okay.
Just a bit. Sorry about that. Before we get rolling here.
yeah, that would be a major continuity gaffe. Suddenly his head's at the top of the screen.
You crew.
And okay.
Yeah, it's great to have this conversation. I'm George Church, professor of genetics at Harvard Medical School.
Thanks. All right, so there are so many things that I could ask you about, but I've narrowed it down to just a few that I think, anyway, have the most to do with prediction of the work that I know about that you've done. But at the end, I'm hoping that we can leave some time for you to tell me what I forgot or what
Okay.
you're doing next.
Yeah.
Okay. So the first time I became very aware of your work had to do with the Personal Genome Project. And I'm not sure if you even were aware of this, but someone from your lab asked us at the IIC, this Initiative in Innovative Computing that I used to run, to help with a system for educating patients online, which of course you now have. Okay.
Yeah.
And so what I wanted to talk about was the Personal Genome Project in the context of what people do worry about in terms of predicting the future of both people who have already been born and unborn people, and then what you think they should worry about. I'll just lay it all out there at once so you can comment on whatever you want at whatever time. And then also the societal implications of having a huge database of genomes. And I know that your project has gone international now, and that in Austria, right? Is that called
Yes,
the
Genome Austria.
Genome Austria, right?
Yeah.
And so it's obviously very tied into what the government is doing and what society should know about not necessarily the path of an individual, but what you can predict about a population. So do you want me to ask you those questions one at a time, or do you want to just muse about those things?
How about if I give it a crack and then
Yeah.
you ask me whatever I
Perfect.
forgot?
Go for it.
Because there's nothing in there that I don't want to answer.
Right.
So
Okay.
yeah. I mean, the Personal Genome Project is a great example of a way that we can collect large data sets about individual people and then share them as individuals, which allows other groups all over the world to reformat and reuse that data in various ways. It's like Wikipedia, but with a lot more raw data.
And just to confirm, everyone who participates is still agreeing to have their genome made public, is that right?
Right.
Through the whole project?
So everyone in the project agrees that all their data can be shared, not just their genomes, but their traits and their trait data and their environmental data. which includes their medical records and things that go far beyond conventional medical records to what we hope will be the sort of data we collect in the future. And so the sort of data that's available for study to anyone in the world, and so this is the only we know of open access human biology data sets, are things like microbiome, proteome, immunome, transcriptome, genome, MRI of the brain, et cetera. And some of this is open-ended, but we don't know where it's going, but we can have a conversation about what predictions are plausible and which of those are highly desirable and which ones cause people to pause and think, are we ready for it?
And the other thing I should note is that in the course, especially in the third part of the course, which this will be part of, we want to really emphasize the quantification of uncertainty and how you explain that to people. And so if you can also emphasize which things we're pretty sure about and which things we are unsure about and how that uncertainty is measured would be great.
Right. So genetics is sometimes represented as being, the thing that's disturbing about it is that it's uncertain. But in fact, a huge fraction of medicine that's not genetic is uncertain as well. And we're particularly poor at representing that uncertainty, and so we tend to dodge it. But with genetics, we don't dodge it. We have good ways of indicating the certainty, even though there are environmental and genetic components, and the environmental part includes medicine, which is itself a moving target. We can still make predictions, for example, based on twin studies of the amount that's heritable. But if there's anything that's also environmentally affected, you can have something that's clearly almost 100% heritable, meaning predictable from your genes. Classic example is phenylketonuria, PKU, which is one of the things that almost every child is diagnosed for on birth from a little heel prick. There are about 40 different genetic conditions that are predicted from the metabolites, or you could say observed, so the prediction is so guaranteed. But even that is not destiny. It's not something that, even though it's 100% genetic and you're essentially measuring the metabolic disorder on the whole point of doing it at birth is you don't want to, typically there's not a high priority for medical predictions if there's nothing actionable. But with PKU and many hundreds of other diseases, there are things you can do. So with PKU, you just avoid phenylalanine in your diet, particularly amino acid. And that changes it from 100% genetic destiny to 100% environmentally managed.
If you do what you're supposed to do.
If you do it, as long as the child and the parents do what they're supposed to do. Now, and I think that nicely illustrates a huge fraction of genetics, and that's why we collect genes, environments, and traits. We sometimes call it GET. Because we want to see the outcomes for people over long periods of time, and to see what we can do to change their environment in order to change that. A new thing that we can do is we can also change their genetics. So gene therapy, there are now 2,400 clinical trials on gene therapy, and so in a way neither the environment nor the genetics is carved in stone when you're born.
Yeah, CRISPR and all of those other associated techniques was on my list for later, but if you want to talk about it now, feel free.
So the important thing when you're talking about what we can predict is it's all conditional, it's negotiable. So the reason we're predicting it is because a huge fraction of them are medically actionable. Now, actionability is an interesting thing, and this is all tied up into how people decide whether they want to do the predictions or not, what they're going to do with them, is you should have a discussion before you go into this as to what don't you want to hear. And to know what you don't want to hear, you have to go through what the options are. Now, even things that you would think are completely not actionable, there's nothing you do, there's no known cure, there's no prevention, it's highly predictive. It seems like you might not want to know that, but there's almost always some kind of actionability. So one of them is you can, if you're going to have children, either accidentally or intentionally, you might want to take that into account. There are all kinds of reproductive options now. So you can impact the next generation. A second thing that you can do in almost every case is you can engage yourself in research. In fact, the Personal Genome Project is all about citizen science and engaging yourself in genetic and medical research. But you can specifically engage yourself in the research on your genetic disease. And I think that, to me,
Which may or may not help you, but it would help future people suffering from the same thing.
It is certainly more likely. I mean, a lot of participation as a human guinea pig is altruistic, but there is also the fact that you're kind of first in line for the breakthroughs if you volunteer your particular disease first, and that helps your whole family.
But there must be a whole interesting psychology of people who do and don't want to know particular things. I mean, I know friends who, when they were pregnant, they didn't even want to know the gender of their child. And I thought, how could you not want to know that? Somebody else knows that, and you don't know that? How could you? And that's me, but that's not how everybody feels, clearly.
Well, there's a wonderful, beautiful range of diversity. Not the sort of diversity we usually talk about, which is like where your ancestors came from, what color your skin, but the brain diversity that is a combination of genes and environment that tell you whether you want to know the gender of your child or even whether you want to influence the gender of your child. So there are now services that is on the continuum with curing inherited diseases that allow you to, before the embryo is implanted, to decide whether you want a boy or a girl. And interestingly, this is surprising to a lot of people, is in the United States, 80% of the decision is for girls. Not so much in Asia, but in the United States, that's the way it is. And so there's the component of prediction that involves changing the future. And I think it's extremely vibrant and alive in the biological and medical sciences.
Right, and that's interesting because that's where we get into all of the controversy, in other words, because just to recap, in the first part of the course, we talk about all of these predictive systems which are almost completely reliant on the idea that there's supernatural influences and that some supernatural powers know something that you don't know, and you're sort of trying to tap into that. And this is almost the reverse. It's like, you know, humans playing God at some level. And there are a lot of people who we both know object to that. But then I know friends whose opinions change when it comes to them personally. So, you know, getting back to the Personal Genome Project, I'm curious if you can give us some examples of people who might have changed their opinion when they started to participate or when they were afflicted with some particular disease or a relative.
Right. So, so there are examples within the Personal Genome Project, even though it's mainly a demonstration project, it's not intended to be give clinical advice.
How many people are participating now?
So there's thousands of people worldwide. It's now in 5 countries. So it started in the U.S. and then Canada, England, Korea. It is Genome Austria, located.
And you're genome number 1.
And I happen to be the first one in the U.S. Because the Harvard Medical School IRB felt that I should have front lines experience to see if there's anything goes wrong, and a couple of things I did catch by being guinea pig number 1. But let's talk about so in my particular case, I learned about my alpha-1 antitrypsin problems. I am a compound heterozygote, which could affect lung. It's one of the major causes of need for lung repair and surgery. But then there was another patient had, I think, a more interesting case. Both these involve predictions. John Lauerman, who I can say his name because he's one of the people who's in the open part of the project. Came in, presented himself as a healthy adult, and then we found that he had a JAK2 mutation, and that JAK2 mutation was probably in his blood. I mean, it was not in his whole body. It was specifically in his blood that it had arisen during his lifetime. So this is a case of your genes changing on their own.
And so is that, would we call that epigenetics, or is that something
That
else?
it is somatic genetics. It is not
Okay.
inherited. So his
Okay, oh, so it's
children,
not inherited, right, okay.
his children would not necessarily inherit that. It's not cancer, but it does involve proliferation, and that's the reason you can detect it.
Okay.
Anyway, it's associated with pain in your peripheral muscles and with scotomas, meaning little dots in your retina. And when we told him this, he says, oh yeah, actually that's on my medical records. I had just forgotten about it. And in fact, he had even explored the possibility it was genetic, and the way genetics was done before the kind of technology that we developed was you would guess at the gene, and then you would do one test for one gene out of 30,000 or so. And if you guessed wrong, then you might try another gene, or you might say, well, I guess that's not it. And it was very primitive. And in fact, generally, you couldn't even get a genetic test, even for one gene, unless you had a clear family history and you knew essentially what gene you were looking for.
Because it was so expensive.
It's
difficult.
expensive, and it was considered that you might get false positives and you
Right.
might freak out. And as it turns out, people tend not to freak out, especially if they've been through the process of understanding what's in the future, and they've what could be in the future, and and that they have the ability to learn certain things and not other things. So that's part of our recruitment and sign up process, is they have to take a short multiple choice exam.
Right.
They have to get 100% on it to make sure they understand what might be predicted and what they and are they okay, if they and their family are okay with
And
those
is that
predictions.
at pged.org? Is that where that exam is, or would there be an example there? I'm just asking because we can link to it from the course.
That's on the Personal Genome Project, although
Okay.
pged.org has some very relevant
Okay.
lessons that are
So not as part of the interview, but we'll send you an email
Yeah,
later
sure.
and ask you what you'd like us to
Yeah.
do. Oh, so what happened in our story?
So John recognized that he had scotomas. He had this leg pain that he thought might be factor V Leiden, a particular variant of a particular gene. When he had done that test before, it had come out negative. In fact, we found that it wasn't factor V, it was JAK2. And so now he takes an aspirin every day, and
That
no
solves
symptoms.
it?
Yeah, and that solved the problem.
Wow.
Right. And there are plenty of other cases of people that have either had surgery or take particular drugs, like the Beery twins. They take nutritional supplements in the dopamine and serotonin pathway. Not exactly clear why those are out on the market, but in this particular case, genetic change was discovered after all else had failed. I mean, they were misdiagnosed as cerebral palsy, a bunch of other very poor predictions, not really based on data,
Right.
or very imprecise, superficial, small data.
Right.
From the big data set, which was their whole genome, or their whole coding part of their genome, they could make this very clear missing gene prediction. And surgeries are often preventative. A lot of people may not understand that it's really a prediction. So when Angelina Jolie announced that she was at risk or that she was getting surgery on breast tissue
Right.
and ovarian tissue, I think some people felt, oh, there must have been a lump that was felt, or a
No.
mammogram that
Right.
showed that there was a lump. It was pure prediction. It was her risk was close to 90% over the next few decades of getting breast or ovarian cancer, and it dropped closer to 5% after the surgery. So these are purely preventative surgeries based on the genome predictions.
And so as an expert, when people ask you, if Angelina Jolie had come to you and said, they're telling me I have a 90% chance, what would you have told her about evaluating how sure the 90%, how certain the 90% number was?
Right. So breast cancer, BRCA1 and 2, is one of the most well established. There was a company already in 1991 that was doing these predictions. Even in that best case, it's not perfect because we don't have the background data on a lot of people who will never develop breast cancer. So it is possible that 87–90% prediction will turn out to be more or less depending on what happens in the general population. That's why it's so important that we have large general population where they're going to, you know, live long and prosper without breast cancer and ovarian cancer. We need that data. But right now, there's a lot of silos that are not sharing that data broadly the way that you share Wikipedia.
And so would you say that that's the most important aspect of collecting a lot of data, is to be able to refine our understanding of various, you know, genetically related conditions for an individual, but based on a much bigger statistical sample?
Right. Yeah, so it's not sufficient that we get, I mean, I think this is one of the best applications right now for collecting large amounts of genome, environment, and trait data. You can't just collect one of those. You can't just share one of those. You have to share all of them, ideally in a big data set per person. If you start to merge them together, it has presumably some advantages, but actually the disadvantages are vast because you're losing information when you do that, even with very clever encryption strategies. So this is one of the big payoffs, is finding out, and it's not so much about deciding whether it's 87% or 67%. It's more, is it 95% under this circumstances,
with the environment
or
traits.
or it's 0% on this other set of circumstances. So it's like, you know, can you change your environment? Can you change your genetics?
So that's like your PKU example.
Exactly.
Okay.
Exactly. I think that's the big thing, because knowing the precise probability, in a way, isn't actionable. I mean, everybody can have their own set point. Each physician can have their own set point and say, and it depends on how onerous is the treatment and things like that. But what you're really looking for are those win-win situations where the treatment's okay, has low risk, and the genetics has high risk without the treatment.
So now I want to ask you a little bit about computation, and we'll get to CRISPR through that. But I know that a lot of the kinds of simulation that we'll talk about in, again, this third part of the course rely on Moore's law. They rely on computers getting faster in a predictable way. And for a while, that's how gene sequencing worked, went along with Jordan. But you like to point out that that's not what happened in the early 2000s, and your lab was quite in large part responsible for that not happening. So can you just explain for the total non-expert, you know, what gene sequencing chips are and why that completely blew open the prediction of how fast we would be able to do that kind of work?
So Moore's law is a really remarkable predictive engine. It's not a law. I mean, it is purely an observation, and it goes much further back than Gordon Moore and transistors in integrated circuits. In fact, it goes back further than transistor. It goes back the vacuum tubes, and it goes back all the way to semaphores where you're communicating with flags. It's this beautiful curve where you get about a 1.5-fold improvement per year, which exponentially accumulates like the best bank account ever. And so, and biology was on that curve too since about the late 70s. It joined the Moore's law, which goes back to the 1800s or something like that. And they're going along, and then suddenly the DNA reading and writing changes slope by about a factor of 5, making it now, and the question is what happened, and what does this mean about prediction? And in this case, it's a very favorable one. The other thing is it could have flatlined.
A lot of people say that Moore's law is something going to end like that because we get
Moore's law could flatline,
limits.
the DNA version of Moore's law could flatline. But in this case, it did something very unusual, which went up. And part of that was because finally the biotechnologists started getting better at being biotechnologists rather than just tinkering around with, messing around with plasmids. And they started getting attuned to the why Moore's law worked, how microfabrication worked. And at first blush, you might think that this revolution was due to either automation or miniaturization. I think those were small factors in that they made things a little more reproducible, a little smaller. But the big thing was multiplexing, which is something that I think even a lot of biologists don't understand what that's all about. But multiplexing was invented for telecommunications by Edison in the 1800s. And it's a way that you can mix a bunch of signals or entities into a shared space and time,
Right.
and then do a lot of processing on them as if they were one thing,
Right.
and then take them apart later, or somehow select the winners later. And this works really in a charming way for chemistry and biology, but there was almost no examples of it before the 1980s. And so we invented this molecular multiplexing and then applied it to reading and writing DNA. So as we miniaturized it, we completely reformulated it so that it could be, so that you could do millions or billions of reactions in a shared chamber.
Right.
And that was what caused the slope to change, and it's still on that curve because multiplexing and miniaturization are continuing on exponential.
Right, and actually a related, not really directly related, but related in my I'm fascinated mind, is I know that recently you've been working on doing gene sequencing in situ, like without actually messing up cells, and then you have these amazing three-dimensional images of the sequences, you know, and in astronomy, I should just tell you that there used to be just imaging. You probably know this, but anyway, there used to be just imaging, and then spectroscopy was invented, which could tell you at any particular point on the sky what was it made of. And then I work mostly in radio astronomy, where for a long time we've done both at the same time. And that is now coming to optical astronomy, and actually the big deal about the James Webb Space Telescope that's going to be launched in 2018 is that it will have these things called integrated field units, or IFUs, which do something which is essentially the same kind of dimensionally expanded imaging that you're doing. So when I saw those images, I was, wow.
You mainly get the z-axis, the depth axis, from redshift.
Well, we do in the distant universe, but it turns out that the same way that a genome isn't a, well, it sort of is a spatial thing, but it's not the same dimension as the two other dimensions.
That's where I was going.
Right, no, but what I mean is you have the image of the genome that's in two actually spatial dimensions, but then you have sort of coordinate along the genome, right, in the other dimension. So in astronomy, we do the same thing. So only in cosmology can we get distance from the Hubble expansion, but in other, like I study star-forming interstellar clouds, and there the spectrum is telling you the amount of stuff as a function of velocity, which is not a proxy for distance in a local. But anyway, it doesn't matter. What's interesting is that the techniques of kind of using more dimensions and then being able to have the computation to process that and understand that in a field as different as mine and yours, it looks the same on the screen. But so how widespread, is that still very experimental, or are a lot of people using that now?
Yeah, so the in situ sequencing is certainly 3- and 4-dimensional, where the 4th dimension is like a spectrum, where every pixel, every voxel in 3 dimensions has a tag that's associated with it, which is a multicolored tag that tells you what molecule is at that point. And very analogous to astronomy, where you have a 3-dimensional object, the universe, and stars have, and other celestial bodies have information, little tags about them, like you know, composition of matter, how fast it's going, and so forth. And so the pictures look very similar. Ours is like a 3-dimensional field of dots with tags associated, and so it is in astronomy. And you can couple it. So the fluorescent in situ sequencing, which is, or more generally in situ information gathering, is a big part, is a growing part of our biomedical, not just research, but biomedical diagnostics. So you want to, there's a company now called ReadCore that manufactures devices and provides a service. So if somebody doesn't want to buy the device, they can just send their samples in. And this is an extension of pathology, essentially, where you'll take a cancer specimen or an epileptic brain specimen or some skin specimen, and you'll want as much information about that as you can get. So you might do serial sections with a microtome, a knife cutting it through, and you can reconstruct very complicated 3D objects.
And is there any sense in which the 3-dimensional arrangement of things is environment in your, in other words, normally you're talking about an external macroscopic environment, but does that microscopic environment matter in someone's genetics? So
both the 3-dimensional structure and the environmental influences on it are extremely important at almost every resolution. So three-dimensional structure is important for the smallest molecules, the bigger, slightly bigger proteins, the folding of the chromosome, the shape of the cell. If you have a cancer, includes an aberrant arrangement of the genes, an aberrant arrangement of the cells. The blood vessels become aberrant, so the tumor will kind of force blood vessels to come in, and they come in a way that's not normal. And all of this is what pathology is about. It's literally the poorly behaving or the abnormally behaving three-dimensional and environmental components of a body.
Right. So I have two forking questions at this point. One is, can we talk about CRISPR and what you can do about editing genes or writing genes? And then the other question is about education. And you and I are having this conversation, and I'm fascinated by it. And part of the reason that I understand it is because we have the same kind of issues in terms of dimensionality and uncertainty and all this in, say, simulating colliding galaxies. But no one's life depends on simulating colliding galaxies. And so when I try to explain this to the public, I don't have to worry. You know, if they walk away and they don't get it completely, maybe I failed, but no one's going to die. And but you and your community does have to worry about that. So anyway, so it's your choice. Would you like to talk about CRISPR now, or would you like to talk about educating the public? Okay.
I'll twist them together.
Okay,
I could twist almost any two topics together. What's peculiar about this particular predictive science is that it's making it into the marketplace. It's making it into people's lives. People have to decide whether they want to participate or not, or how much. They have to decide whether they want to change their destiny by environmental changes or genetic changes. Some of the ways they can change the genetics is through a revolutionary new set of technologies. CRISPR is the one that gets most of the credit, but I actually think the credit should be spread out more broadly to include, you can't edit, CRISPR is an editor of DNA that you can apply in patients, but you can add or subtract DNA or alter it. CRISPR is particularly good at subtracting, but there were previous methods for adding and altering. You can't really alter or edit without reading. So the revolution in being able to read genomes, whether it's in 3 dimensions or 1 dimension, that is the big enabling thing, and that's the drop in cost and improvement in quality and prediction is a key component of deciding what you want to CRISPR, what you want to change about your genome.
Right. And then, so there are people who, you know, and I'm saying educated people who, the same way that they ask me what's the difference between a star and a planet, right? They ask you what's the difference between a gene and a chromosome, right? So if let's assume that you have an interested, educated person, but who just knows nothing about biology. So how do you sort of consent someone to the point where you think they understand when you tell Angelina Jolie, you know, we think you have a 90% chance of getting breast cancer someday. So how do you go about, what is that educational process about getting them to understand enough about the science so that they know what that prediction means?
So we have a lot of people that are not scientists volunteering in our project, and in fact expressing curiosity worldwide whether or not they're in our project. They are so driven that they will eventually learn as much as we know. There are other people who just want to participate. They're either altruistic, or they have a family disease, or they just think it's the right thing to do with their spare time. And for them, we want to give them the minimum information that is as clear as possible about what the risks and benefits are. They don't necessarily need to know that there are 6 billion base pairs in their genome, which is made up of 23 chromosome pairs, half from their mother, half from their father, and that there's G's, A's, T's, and C's. They can learn that when they get interested. But up front, what they need to know is that participating in any medical research has a very high probability of data escape, just like think of WikiLeaks and other hacking. And once it escapes, it has a high probability of being re-identified. And that's not necessarily the biggest risk you'll ever have in your life. I mean, but it is something you should consider. Those are the main risks. And whether there is genetic Information Non-Discrimination Act, it's an act in the United States, it's not a perfect protection, even for employment and medical insurances, which we aim for, and it's not international. And we try not to reassure on that point. You should just decide that if the data got out, you're comfortable with the possible outcomes. And we should all be taking action to make sure that the stigma of particular diseases and economic impact should be minimized, especially for people who are altruistically engaged in medical research.
Because you don't want them to stop.
We want to encourage more and more people to participate, and that means that their benefits to them, their family, and society has to greatly outweigh the the risks, even if they're modest risks. We want them not to add up to something significant. So I think that's what we walk through, and in a way we simultaneously educate and test. They have a fairly short multiple choice questionnaire exam that walks them through each of these decisions, and they can't get 100% on the exam unless they understand the material.
So
and this is in contrast to typical consenting, where they're presented with 12 pages of legally formatted, that's to protect the institution, not the person, and it's all in legalese, and only an attorney, even certain attorneys, can understand, and people typically sign it without understanding, and the researchers often know that. We were uncomfortable with that. We feel that this person should understand what they're getting into.
Computer privacy is a subject where that would be such a better practice than what goes on now. I mean, people don't even understand their Facebook settings, but that's a whole different conversation. So there's two last things I wanted to ask you in the time we have remaining. One is a biology question, and one is just a question about the future, or one of the things I know about the future. So the one about the future is I'm curious about the BRAIN initiative and what you'd like to say about connections between that and prediction. And then the other one is, this is a very, for you, simple biological question, but I bet you that a lot of people who even know a little bit of biology don't know this. So let's say you use CRISPR or one of the other many techniques to rewrite something about the gene. What are you actually doing? In other words, you have some device and you take some goo and you do something to it, and then do you put it back in the person's body? How does it permeate their whole system and have an effect?
Right. So BRAIN and then CRISPR.
Sure.
The BRAIN initiative is a wonderful project that they helped start around 2011, 2012. It's somewhat analogous to a piece of the Genome Project, which was focused on technology. Unfortunately, not the whole thing was on technology, but here the BRAIN initiative has a big technology component. It's baked into the acronym. Innovative Neurotechnologies is the I and the N of BRAIN. And it has a predictive component which is, given a large data set, can we predict what a brain would do, or can we predict how an altered brain would behave? But what we really want to do is collect activity data, which is how the brain is functioning in time and space. Behavior, which is sort of what's the interaction with the environment. What are you sensing, and how are you behaving? So the sensors and actuators of robotics. And then a connectome, which is how the brain is wired up. And that's where the activity goes through that connectome. The development, which is how that it came out of a single fertilized egg. Single cell became this amazing, one of arguably the best computer in history all over the world. And then the expressions is how the genome that resulted in that developmental biology, how that simple 6 billion base pairs that you inherited turns into this very complicated computing device inside our skulls and actually throughout our body. So we're measuring these things: A, B, C, D, E, F, activity, behavior, connectome, development, and expression. We're collecting those in a systematic way, starting with small brains, say
Not people's small brains, right? Animals' small brains.
Like mouse embryos are
Right.
smaller than adult mice.
Right.
Adult mice are smaller than many other animals and humans. But there is some human data collection being done. You can do MRI, which is low resolution. You can do brain organoids. You can do samples from living people that have epilepsy or autopsy samples. So there's a lot of different sources of, and this is definitely big data if there
And
ever
is there
were
some
big
sense of
data.
ever connecting like the architecture that you get from the connectome with that kind of 3D in situ sequencing?
Right.
Would that
So the
happen?
way that our team does the connectome is, in fact, we can do essentially all of the A, C, D, and E by in situ sequencing. The activity is
On brain tissue.
on brain tissue.
Yeah.
Yeah. So the activity is typically measured by real-time calcium imaging with a fluorescent, where you use a 3D microscopy, but it can also be encoded into DNA, and we're working hard on making that work better. But then the other ones, we're just reading out either barcodes, the same way you get kind of like the spectral signature for each RNA type or each
Right.
connection type. All of that is built in a 3 and 4-dimensional model.
That's incredible. Okay, so now my easy biology question, and then we have to let you go.
Yeah. The CRISPR one. Yeah.
The
You wanted me to say how do you actually do CRISPR?
yes, right.
Yeah,
The CRISPR.
right.
Well, in other words, how does it get implemented in a patient?
Right.
Yes.
So there are 2,400 gene therapies approved for clinical trials. Some people think that it takes 10 years before anybody gets cured. Well, it takes 10 years to get full approval to be sold, but people get cured in the first year of a clinical trial if it's a really great therapy. Anyway, there are many gene therapies. Most of them, like 99% of them, don't involve CRISPR or any other editor. But in all of them, they have in common is you either treat the patient without taking cells out or you take them out. So you might take T cells out from an HIV patient, treat them so that you delete the gene that encodes the receptor that the virus gets into the cell. So basically you take the T cells out, you with electricity get the DNA in. The DNA will then set a set of nanomachines that will go in and edit the CCR5 gene.
Nanomachines, just to be clear, natural, organic.
These
nanomachines.
are engineered proteins and RNAs, which constitute an engineered nanomachine that is based on natural things that we found in bacteria, but it's very unnatural by the time we've turned it into a therapy.
Got it. But they're not like little metal robots.
They don't have much metal in them. They're mostly made out of carbon, nitrogen, oxygen, sulfur, and phosphorus, and hydrogen. Anyway, so these organic chemical nanoscale catalysts that you've programmed in the computer to be able to target one place in the 6 billion base pairs, they will go in, they'll cut that site, and then they'll make it so that now the T cell is resistant to HIV. You put the T cells back into the patient, and now the patient gets cured of AIDS. With blindness, it's a slightly different procedure. Here, you can't take the retina out conveniently, so you inject into underneath the retina, and now instead of electricity, you might use viral particles. Viral particles will encapsulate the DNA or RNA, and it will bring it to the correct position. And then again, the capsid brings it into the cells. The cells get reprogrammed by these protein RNA nanomachines, and now the person is cured of blindness.
Right. So it seems like the whole path is to figure out genetically what's going on, and then to figure out some way to deliver whatever that solution is.
So in both cases, you make a prediction from their genome. You say, oh, this person has HIV, and they also have a genome that's sensitive to HIV. Let's change it so they're resistant to HIV.
Right.
Oh, this person is blind, and they have a particular genetic change that makes them different from everybody who's not blind.
Right.
And so you predict that if you change that, they will not be blind anymore. And both of these predictions turn out to be true, and they become therapies.
That's excellent. So we're going to make you late if you stay
Yeah.
any longer.
Yeah.
What
Yes,
time is it?
It's 11 o'clock.
It's 11 o'clock exactly.
Okay. Yeah.
What, was there anything that you wanted to say that I didn't ask you?
No, you covered it. Well, I mean, I could go on for hours.
I know, but I don't
many-
want to make you
many
late.
Tens of
They
hours. But why don't we do
particularly
that as a sequel?
Okay, sure. We could do
I
that.
mean, I love what you're doing with this project, and
thank you.
I think that a lot of things we do are involved
They're very,
in prediction.
very related, yeah.
And they're predictions that people should care about. What's amazing, actually, maybe this is one thing that is worth adding,
Yeah.
is that well-educated, wealthy individuals that are curious and so forth, for the most part, have not had their genome sequenced, even though the price has come down from $3 billion to $1,000.
I haven't. I volunteer.
And that means that they either are not curious and they don't realize how valuable it is
Right.
to everybody. I mean, there's no person who knows in advance that there's nothing there we'll learn from the genome. There are people who will learn nothing from the genome, but you don't know that until you look at it.
You know what?
right?
I can't believe I've never done that. So as my thank you to you,
Okay.
I will officially
Okay.
join the project, and we'll keep
Excellent.
that in the course.
Okay.
Okay.
Thank
Wonderful.
All right, and we'll see
Thank
you
you.
again soon.
Yeah.
I think we should talk more. So this,
Okay.
this is really fun.
Great.
So I'm not going to Sci-Fu this year. Are you going?
I don't think so, no.
Yeah.
Yes.
I'm not
All right,
going
we need
to.
30
So
seconds.
all we need is
Oh, what do
just-
we need?
Yes,
All we need is just
Quiet
the,
room.
sound of the room, so 20 seconds of silence starting now.